Hello all, today we will be discussing microbial transport. Microbial nutrient transport is the uptake of different nutrients required by microorganisms for growth. The first step in nutrient use is of course the uptake of these nutrients from the environments to the microbial cell. Different mechanisms are used for taking in the nutrients as per the cell requirement. These uptake mechanisms must be specific. Only the necessary substances must be acquired, because a cell does not benefit from taking in a substance that it cannot use. There is no point spending energy or accumulating substances which it does not use. Now, microorganisms often live in diluted environments, nutrient poor habitats, and they should be transporting the nutrients into the cell against a concentration gradient. Very often the amount of nutrients in the external environment will be very less compared to that which is present inside the cell. So the nutrients must be transported against a concentration gradient from an environment which is less in those nutrients to an environment where it is present in higher concentration. And the nutrient molecules must also pass through a selectively permeable plasma membrane. It does not permit free passage of all substances. Most substances are inhibited by this plasma membrane. Because of the enormous variety of nutrients and the complexity of uptake process, there are different uptake mechanisms, transport mechanisms used by microorganisms. Most important being active transport and passive transport. Now, passive transport includes mainly diffusion and facilitated diffusion. Diffusion, as we all know, is the movement of materials across a selectively permeable membrane according to the concentration gradient and without the help of carrier molecules. In facilitated diffusion, carrier proteins may be involved, but generally diffusion is without the help of any carrier molecules and according to the concentration gradient across a biomembrane. Diffusion is a slow process where molecules like water, oxygen, carbon dioxide and small sugars pass across membranes. They move passively from the region of their higher concentration to the region of their lower concentration till an equilibrium is reached. Once an equilibrium is reached between the external environment and the internal environment, there is no further transport of nutrients. It can take place in both directions and no energy is used for diffusion. So nutrients can come inside and nutrients can move outside using diffusion. No energy is used for this process. So this is according to the concentration gradient, usually does not involve carrier molecules. It's a slow process. It moves from the region of higher concentration to region of lower concentration till an equilibrium is reached. This takes place in both directions and no energy is used in this process. Active transport, whereas, is the transport of solute molecules to higher concentrations from lower concentrations, or it is usually against a concentration gradient and with the use of metabolic energy. Active transport involves specific protein carriers. It differs from facilitated diffusion. Facilitated diffusion also uses some carriers we had mentioned, but active transport differs from facilitated diffusion in its use of metabolic energy and its ability to concentrate substances. In facilitated diffusion, though carrier protein is used, there is no energy involvement, and there also the transport happens only till equilibrium is reached, whereas in active transport there is use of metabolic energy and substances can be concentrated. The transport need not stop once the equilibrium is reached. Active transport is a rapid, fast process. It takes place only in one direction. It enables selective uptake of materials like proteins, ions and complex sugars. So these are the differences between active transport and passive transport. In the diagram we can see diffusion, where molecules move inside the cell, facilitated diffusion where a carrier protein is used, and active transport where energy is involved as well as a carrier protein is involved. Now, little more elaborate about diffusion. A few substances such as glycerol can cross the plasma membrane by passive diffusion. Passive diffusion, often simply called diffusion, is the process in which molecules move from a region of higher concentration to one of lower concentration because of random thermal agitation. Agitation can help this rate of diffusion. Rate of passive diffusion is dependent on the concentration gradient between a cell's exterior and its interior. So, diffusion occurs according to the concentration gradient from a region of higher concentration of nutrients to a region of lower concentration. A fairly large concentration gradient is required for adequate nutrient uptake by passive diffusion. That is, external nutrient concentration must be sufficiently high for the nutrients to be transported into the cell. Now, the rate of uptake decreases as more nutrient is acquired unless it is used immediately. As more and more nutrients are acquired, it reaches equilibrium inside the cell and outside the cell. So, if the nutrients are not removed or are not used immediately within the cell, the rate of further uptake may decrease. Very small molecules such as water, oxygen and carbon dioxide often move across membranes by passive diffusion. Larger molecules, ions and polar substances do not cross membranes by passive or simple diffusion. Only very small molecules can easily move across by passive diffusion. So, that was about passive or simple diffusion. Now, we have facilitated diffusion. Facilitated, the term means assisted. Facilitate means to assist or to help. So, facilitated diffusion is carrier aided diffusion. Diffusion with the help of certain carrier molecules. The rate of diffusion across selectively permeable membranes is increased if carrier proteins such as permeases, which are embedded in the plasma membrane, are employed. So, carrier proteins such as permeases, if involved in diffusion, the diffusion rate is increased across selectively permeable membranes. Now, carrier proteins are specific or selective and will transport only closely related solutes. So, they are specific. Carrier protein is specific for specific solutes or specific nutrients. Rate of facilitated diffusion also increases with the concentration gradient. It is similar to that of diffusion. Rate increases according to the concentration gradient. Only difference between diffusion and facilitated diffusion is that carrier molecules help diffusion and facilitated diffusion. Other things are pretty common. Some examples of carrier proteins: aquaporins transport water, glycerol facilitators help in glycerol diffusion. Thus, facilitated diffusion is truly diffusion through a carrier which is involved in the process. Movement of molecules is according to the concentration gradient across the membrane and no metabolic energy is required. If the concentration gradient disappears or an equilibrium if it is reached, there is no net inward movement. There is no further movement of nutrients. So, how can we maintain the gradient? Gradient can be maintained, as we discussed before, by utilizing the compound or by transforming the transported nutrient to another compound or by moving it to another membranous compartment. This can happen in eukaryotes where different membranous compartments are available. In prokaryotes, it should be either utilized or it should be transformed to some other state of that compound so that the gradient is maintained. So, if we represent the diffusion rates of passive diffusion and facilitated diffusion, we will get the graph like this. In passive diffusion, the rate of transport starts from low and it goes on increasing according to the substrate concentration till the concentration gradient is maintained, till there is more substrate outside the cell and less inside the cell. Whereas in facilitated diffusion, as time goes on, the carrier becomes saturated. Carrier protein has binded with and transported as many solute molecules as possible. Beyond that it cannot bind. So there is a plateau effect above a specific value, and that is seen in case of facilitated diffusion. In passive diffusion, till the concentration gradient is maintained, the graph is linear, steadily increasing till the concentration gradient is maintained. But in facilitated diffusion, after a value, the carriers have become saturated and they cannot be further transported. So it reaches a plateau after a point. So this is the rate of transport with or against concentration of nutrients in case of diffusion, passive and facilitated diffusion. Plateau effect is seen in facilitated diffusion because of the saturation of transport membrane proteins. Now, what exactly happens to the carrier protein complex? As you can see in the diagram, the carrier molecules, first the solute molecule binds to the outside of the carrier, then the carrier changes the conformation, it releases the molecule on the cell interior. The carrier would then change back to original shape and is ready to pick up another molecule. So the carrier protein complex is seen in the cell membrane, and first in the initial conformation solute binds to the carrier, then the carrier changes the conformation and it opens up towards the interior of the cell, and to the interior of the cell it releases the nutrients. It again changes back to the conformation and it opens up to the exterior of the cell. More solutes can bind to it. So this goes on in the facilitated diffusion process. Because there is no energy input, molecules will continue to get transported only as long as their concentration is greater on the outside. Thus, a lipid insoluble molecule can enter the cell in response to its concentration gradient through the carrier. Though it cannot enter through the membrane, it can enter through the carrier, lipid insoluble molecule. Mechanism is driven by concentration gradients and is reversible. If the solute's concentration is greater inside the cell, it can move outward through the same method. If the cell metabolizes nutrients upon entry, influx again goes on continuing. If the cell removes the nutrients, the gradient is maintained, influx is favored or it goes on. Facilitated diffusion is not very important in prokaryotes because nutrient concentrations often are lower outside the cell, so that facilitated diffusion cannot be used in uptake. Where it is handy is in case of glycerol, which is transported by facilitated diffusion in E. coli, Salmonella typhimurium, Pseudomonas bacillus and many other bacteria. The process is more prominent in eukaryotic cells where it is used to transport a variety of sugars and amino acids. Thus, microorganisms, as we saw, often live in habitats characterized by very dilute nutrient sources, and facilitated diffusion mechanisms are not adequate to transport and concentrate these nutrients because here the transport is across or according to the concentration gradient, and that is not always feasible in case of microorganisms inhabiting dilute nutrient environments. In such cases, microorganisms depend upon active transport and group translocation, which are both energy dependent processes to transport nutrients. That we will be discussing in the next class. Hope you have understood the passive transport. Thank you.